Okay, let's move on to part 3 of chapter 9. So, um, sometimes a trait that is dominant doesn't necessarily mean that that's the most common one. As you can see, when we went back and talked about the freckles, right? Freckles aren't actually that common. Not many people have them. And so, but yet the freckles is actually dominant. The having of freckles is actually dominant. So what we call it usually um, is we refer to dominance is basically more of a way that says that if you have a heterozygous genotype, so one big, one small, what you get is the result that you get, the phenotype is the dominant phenotype. And so we like to refer to it as a wild type. The wild type is the one that is like oh sorry, no, we don't like we like to refer to the one that is most commonly seen in nature, not as a dominant one, but as the wild type phenotype, so wild type traits. And these are not necessarily specified by dominant alleles because, like I said, dominant doesn't necessarily mean the one that shows up the most. Because freckles means dominant, non-freckles means not dominant, and freckles, not having freckles is actually what we have the most often. So part 3 is all about the genetics of basically human characteristics. And even from what we just talked about, the dominant not necessarily being the one that is most common, you can understand that human genetics is actually quite complex. So eye color, hair color, they're very complex and actually poorly understood. Not many things follow simple Mendelian genes. So not many things have the dominant versus recessive thing. Sometimes we get things that are even more difficult. There are some things like freckles or having a widow's peak, which I don't have, but like having a widow's peak on your hairline that you get if you have like two dominant alleles, or if you don't have if you have two recessive alleles. But then there's other patterns of inheritance that are very, very complicated. Okay, so first we'll talk about some things, patterns of inheritance that in humans that do follow normal Mendel genetics. And then we'll talk about, I think, yes, we'll talk about things that don't really follow normal Mendel genetics that are a little bit more complicated. So a lot of disorders, actually, a lot of genetic disorders are disorders that basically follow normal Mendel genetics, meaning if they're dominant or like two dominants or two recessives or one dominant, one recessive, etc. And if we have one dominant and one recessive, the dominant is going to show up. So there's lots of different disorders. They could be either recessive disorders, so those are disorders that where you need both recessive alleles, or they could be dominant disorders where you just need one dominant allele. So some of the examples of recessive disorders are things like albinism, so being albino, having cystic fibrosis, those kind of things. And I believe we already actually kind of talked about cystic fibrosis, but basically all of their fluids are quite viscous. And so it makes so like any kind of mucus that they have, very viscous and it gets stuck in their lungs. They have troubles breathing and they can easily get infections, right? Being an albino, obviously, it's like lack of pigmentation. So some of these, in order to get these, you have to have two recessive alleles. So for you to have two recessive alleles, one recessive has to be from the mom and one recessive has to be from the dad. But not necessarily do the mom and dad have to have the genetic disorder. They can simply just be carriers. While dominant disorders, you're going to have at least one dominant allele. And it's actually interesting. So something like dwarfism, for example, you only need to have one dominant allele. So if you do not suffer from dwarfism, that means you have two recessive alleles. So just again, the dominant isn't necessarily always better. Um, okay, so most genetic mutations that cause medical disorders are recessive, as you see here in the table. These are all the recessive ones, and there's so many more. And then here, there's only like 4 dominant disorders, so where you have to have a dominant allele. But most of them are recessive, meaning you have to have 2 recessive alleles. Individuals who have the recessive allele, but they themselves don't actually have any of the symptoms of the disorder, are known as carriers. So these individuals are heterozygous, meaning they are like big A, little a. Okay, so they have one recessive allele, but one dominant allele, right? And because they have that one dominant allele, they don't actually have the recessive disorder. But if they come together with another person that has the recessive allele, then they have a 25% chance of making a kid with, like having a child with the recessive disorder. Cystic fibrosis is one of the most common ones, and it's also quite lethal as well. There's some statistics here about 1 in every 31 Americans or 10.5 million Americans are carriers. And so we can use Mendel's laws and even the pedigree to see if we have it, if it could be passed on to future generations or not, if you are a carrier. So a carrier, just to give us a definition, it's a person who does not suffer from the disorder, right? But who carries the recessive allele so that it can be passed on to the offspring. So here you have the example of albinism I was giving. You have parents which are big A, little a, big A, little a, so they are both normal in pigmentation. But when they come together, you can see that there's 1 out of 4 or 25% chance that they have an albino kid. With dominant and dominant disorders, these are this is basically, again, a genetic disorder that comes about if you have the dominant allele for this particular gene. So an example is achondroplasia, which is basically dwarfism, where your head and torso develop normally, but the arms and legs are short. And here you have to have either a homozygous dominant will give you, will give you achondroplasia, or heterozygous, so one dominant and one recessive. This also means that if you have achondroplasia, you have a 50% chance of passing it on to your children. So you could do a little Punnett square of like thinking of someone who is heterozygote, right? And comes together with a person that doesn't have any. Like, okay, actually, let's do it together. So let's pretend we have an individual who has achondroplasia and is a heterozygote for achondroplasia. So big A, little a, right? And they come together with a person who does not suffer from dwarfism. And so if that happens, actually, sorry, the genes, the gametes are going to be big A, little a here. My pen is finishing, sorry. Big A, little a for this person that has dwarfism. And then this person is going to have little a, little a. Okay, so let's make the Punnett square. Big A, little a here, little a, little a here. So the big A is going to be passed on to these two possible offspring. And then here we're going to get the little a from them. And so because of this, if you have achondroplasia, 50%, so 2 out of 4, 50% of your children may probably develop achondroplasia as well. That's basically what that was saying. Okay. Oh, I didn't realize it was, there we go. Instead of saying A, they used the letter D in this case. All right, so dominant alleles that cause lethal disorders are much less common, and this is because they don't really get a chance to grow up past puberty to pass along their dominant alleles to their children. There are some examples of some particular disorders that are lethal, but at a later stage in life and not at an earlier stage in life. And this is an example of is like Huntington's disease. So Huntington's is just a degeneration of your nervous system, and so like you have neurological problems, and until like it starts usually, but not always, in middle age, and then you have a few years before you pass away. And this is also something that you get if you have the dominant allele. But at this point, since it starts at middle age, people usually have already procreated at this point, and so there's a really high chance, 50% chance, that that their children also have the particular bad dominant allele. So it's a really sad disorder. Okay, so Mendel's two laws explain inheritance in terms of genes that are passed along from generation to generation according to simple rules of probability. And these laws are valid for all sexually reproducing organisms. So the law of segregation and the law of independent assortment apply to us as well. But Mendel's law stops short of explaining some patterns of genetic inheritance. So some patterns are very complex. Like eye color, we don't only have 2 variations of eye color, we have a lot of variations of eye color. Height, we don't only have 2 heights. We don't only have like people that are 5 foot or 6 foot, right? We have all sorts of variations in between and even into the extremities. We have all sorts of variations. So especially when you think about humans, we have a lot more complex kind of inheritance patterns. So one of these, so this is stepping away from Mendel. His laws are still applicable. So law of segregation, our alleles are still being segregated in their gametes, and law of independent assortment, when they do segregate, the alleles just go up randomly. Those two are still valid. However, it's no longer the simple dominant versus recessive thing. It's no longer that. So let's look at the first example. The first example is incomplete dominance and codominance. So with incomplete dominance and codominance, this is when you have, yes, you have a dominant allele and a recessive allele, but when they come together, when they're paired up, they actually mix, they blend. Okay, so for example, they blend in the sense that both of them are expressed, both of them give off their color. And so when you have something that is red and something that is white, they come together and they form something that is pink. That being said, it's not proper blending, like it's not actually blending two colors, like two paint colors. If I was to take red paint and white paint and I blended them, I get pink. There would be no way, no matter how many times I pair it back again with some more white paint, I could never make that pink paint completely white again. So in this case, it's not like blending, because if I have an R, big R, little r, which in this case is incomplete dominant, so that's why it's not fully red. If I have this and I pair it together, so a pink flower with another pink flower, big R, little r, if I have two little r's, I can get my white again. And if I have two big r's, I can get my red again. Do you guys see that? So if it was true blending, the true red and the true white would never show up again. But as you can see here, they do show up. So so basically it demonstrates that the red and white alleles have not irretrievably blended into pink. Rather, they have come together in F1 only to separate again in F2. So in incomplete dominance, you have basically the thing that the allele, the protein that the allele encodes is expressed, is made. So think of the genes as they encode a protein, right? And so this big R is going to encode the red protein. This little R is going to encode the white protein. So red plus white come together, we have pink. This little r here, if I have little r, both of them are going to express the white protein. So white plus white, we have white, if that makes sense. So neither the red nor the white allele is completely dominant. Each has incomplete dominance. Incomplete dominance is a genetic condition in which the heterozygous phenotype is distinct and the intermediate between either of the homozygous phenotypes. Okay, so homozygous dominant in this case is red, homozygous recessive in this case is white, and then the heterozygous between the two is going to be pink. We also have codominance, and this is when basically both alleles are dominant, and so both of them will be expressed. Neither one is going to trump the other one. So this is, you can see this with cows. Actually, when you see a cow with like spots, you know, your typical cow that makes milk kind of thing, you see it like white with like black spots and everything. That's because it's both expressing the black allele and the white allele together. Both alleles, there are just two of them, both are going to be expressed. Neither one is going to trump and shut the other one up. Okay, so that's codominance. Another example of this is our own blood type. You've probably heard of the ABO blood group in humans. We actually have three alleles in here, three possible alleles, but in a human, we always have two per gene, right? So we have one allele gives us the A, so IA, then we have allele B, IB, and then we have little I, which gives us the non-functional one. You're like, okay, so this makes no sense, but it will make sense right here. So if someone has IA and IB, okay, both of them are dominant, both of them are going to be expressed, and so you actually have a person with AB blood type. You see that? If you have IA, both of them are A's, you're going to get a blood A. Or if you have IA little I, which is non-functional, it doesn't work, you're also going to get, since you have IA here, you're also going to get blood type A. IB or IB little I, you also get blood type B. The only time you get the O is with two little recessive I's, okay? And that's when you have blood type O. So you can see that if I have IA and IB, which are both dominant, you can see that IA is dominant to little I, IB is dominant to little I, so it becomes B to blood type. IA to dominant to little I, it becomes a blood type A. If I have both of these, I get an A and a B blood type, so I have blood type AB. If I get none, if I only have my recessive, then I get my blood type O. So codominance is basically where both alleles are expressed, both of them are dominant, neither one is recessive. And so in the case of IA, IB, both are expressed. We also have pleiotropy, and pleiotropy is something not too difficult to wrap your head around. So until now, we've talked about one gene affecting only one phenotype. Okay, so the eye color affects only the eye color gene affects only your eye color your eye color phenotype. However, a lot of times, most genes actually have multiple effects, so multiple physical phenotypic effects, and this is known as pleiotropy. Okay, pleiotropy is when one gene influences several characteristics. An example is with sickle cell anemia. So sickle cell anemia, we've mentioned it already before in past chapters, but again, it's a genetic disorder. If you have little s, your hemoglobin does not properly form a correct structure, and so your red blood cells look like this. This is not what your red blood cells normally look like, right? So this is the first phenotypic change that having that particular gene does, right? So having little s, you get your red blood cells to look like this. In addition, though, this is where we have multiple effects, right? In addition, because your red blood cells look like this, these red blood cells are more likely to clog up in your veins, and therefore you are more likely to suffer from a heart attack. So there's a second effect. Because of this also, in addition, because of this this red blood cells looking like this, they aren't very good at keeping oxygen. And so because of that, you're also anemic. So there you have already 3 effects. So you can see that one gene can, especially in humans, have more than one effect. So this particular gene doesn't just affect the shape of your red blood cells, it also affects how often you have a heart attack. It also affects if you're anemic or not. So it affects many different kinds of properties, physical properties. Okay, so that's pleiotropy. Then we have polygenic inheritance. Polygenic inheritance is basically when more than one gene is going to contribute to one specific phenotype. Okay, so it's the inheritance of a genetic character that is determined by the interaction of multiple genes, with each gene having a small additive effect on the character. An example of this would be human height. We don't have just one single gene that tells us how tall we are going to be in our adult life. We have many, many genes. For example, you probably have the gene that is involved in bone growth. You probably also have a gene that is involved in obtaining nutrients from your food, right? The better you are obtaining nutrients, the faster you grow, right? The better you grow, the quicker you basically get to your prime. And so there's many genes involved in actually taking into consideration just one specific physical appearance, and that is your height. And when that happens, that's known as polygenic inheritance. And usually you can see polygenic inheritance because it gives you so we see this continuous variation where you have usually a lot of people that are average height, right? They're the ones that really make up the bulk of the population. And then people that are a little bit shorter or really, really tall usually are found in the extremities, and they're not seen as often. Okay, so this is where we get that continuous height. In this case right here, this is the beak depth of the bird. You also have, birds usually have an average size beak compared to like a smaller or a bigger one, right? So that's basically polygenic. So multiple genes affecting one specific characteristic. And that's actually what we have with eye color. That's what we have with hair color, with skin color, with height. There's a lot of different kinds of like characteristics, like phenotypes on humans that are involved with, that have polygenic inheritance. So more than one gene is involved in actually giving the final outcome. Another thing to consider is basically epigenetics. And epigenetics is essentially when the environment, so it starts with E, right? The environment plays a role in the genetics, right? So a really simple thing, and this is kind of silly, so you have twins, one of them dyes their head black, their hair black, and the other one is like normally like a red-haired person. So obviously they look different from each other, and that's something that the environment is doing, right? She dyed her hair black, so her hair is not black because her genes tell them that they're black. It's not like that. So that's a very silly example. Another example would be you have darker skin color because you go out in the sun, right? That's not something that like in the sun kind of burns you and kind of gives you a tan and changes the color of your skin a little bit. So you have to realize that the environment does play a really big factor in stuff. So it's not just your genes. And so this is known as the nature versus nurture effect. And so the examples that I've been giving, I gave you guys, are very silly examples, but sometimes the environment really does go ahead and actually physically change your DNA. So even if you are a twin, right, and you're identical to each other, the environments that one twin is exposed to can change the DNA just a little bit from the other twin, and they can be a little bit different. And so this is why we had studies where between twins, one has schizophrenia and one doesn't, right? So there's a lot of things, and this is all because of epigenetics, or basically the role of the environment on the actual genes. So that is basically, no, not it. We still have a little bit ways to go. The next thing we're going to talk about is the chromosome theory of inheritance, and that just basically is a fancy way of saying that your genes are located on your chromosomes. This is there's a whole theory behind it because, sorry, I have to fix it. There's a whole theory behind it because initially people didn't know that your genes lied on your DNA, or they were found on your chromosomes, that your chromosomes were made out of DNA. People did not know. So they have to carry out a variety of different kinds of experiments to come to this one conclusion, that on your genes you have, on your chromosomes you have your genes, right? An allele per homologous chromosome, and each specific gene has a specific location known as a locus, which we already talked about. Okay, and by knowing this, we can also basically relate these genetic patterns and the law of segregation and the law of independent assortment back to meiosis, because meiosis works with chromosomes, right? And so this is where it all kind of stems from. So another thing that we have to talk about is linked genes. So linked genes are basically the genes that are found next to each other, side by side on one chromosome. So this means that if I like, I'm actually going to do a thingy, sorry, draw. I'm going to go back to drawing. So if I have chromosome 1 right here, right? Chromosome 1 right here with this gene that gives me blue eye color. Okay, and then I have another gene right next to this, like right next to it, and this gene is for I don't know, to have freckles. Okay, it's one of the alleles for freckles. So one of the alleles for blue color eyes, and then one of the alleles for freckles. These genes are going to travel together. Okay, if once we have, and this is my homologous chromosome 1, okay, and say this is still eye color, but instead of blue, it's green, and here I have my freckle gene, right? But instead of having freckles, being dominant for freckles, it is recessive for freckles. So because these are next to each other, when they segregate and they go to different gametes, right? They segregate. The blue and the positive freckles, being with like the ability to have freckles, are going to come together, right? And so this kind of goes against what Mendel figured out with the law of independent assortment. But this is just because your genes, in this case, are physically near each other. I can't take this chromosome, isn't going to be broken up or anything. The whole chromosome is going to be put on another gamete, on like one gamete. So because of that, linked genes actually travel together. Okay, so linked genes are often inherited as a set, and therefore often do not follow Mendel's law of independent assortment. So far, the patterns of genetic inheritance we discussed have always involved genes located on autosomes, right? So these are chromosomes that are not involved with sex. So these are chromosomes 1 through 22 in a human, and we haven't talked about sex chromosomes. So that's another thing that we have to talk about, in addition to everything else, is how inheritance works with sex chromosomes, because it is a little bit different, especially when you think about males, right? So males, their sex chromosomes are an X and a Y. That means that they do not have a homologous pair. X chromosomes are very different looking than Y chromosomes. You can see it here in this picture. X chromosomes are really bulky. The Y chromosomes are teeny tiny. They're not homologous. Neither of them carry the same types of genes. They're very different from each other. And so because of that, the inheritance patterns for males are, like when it comes to sex chromosomes and the genes that they carry on the sex chromosomes, very different from what females have, because we have 2 X chromosomes and we have normal homologous chromosomes when it comes to that. So if a male has So for example, there are a few genes that are found on only the X chromosome and not on the Y chromosome. Like for example, the red-green color blindness, okay? So the genes that are found on sex chromosomes are known as sex-linked genes. And an example of the sex-linked genes is the ability to see color. Most males do not see, or not most males, sorry. Usually males are, they're more, goodness, I can't talk, sorry, red-green colorblind males than there are red-green colorblind females. In fact, there's about 8% of the male population is red-green colorblind, while 0.02% of the female population is red-green colorblind. So this, I keep pointing to this picture right here. This is because in this picture, there's a number, right? And I can see the number. It's actually, maybe you guys have seen it too, it's actually the number 7. If a person is red-green colorblind, they cannot see this number because they cannot see the difference between red and green, okay? And so why is it that males get red-green color blindness more often than females? And that's because this particular gene to see the red and the green is found on the X chromosome, okay? So it's found on the X chromosome. So because it's found on the X chromosome, females have 2 slots. So if we have the dominant ability to see the red and the green, and we have the other one is non-functional and is recessive, we can still kind of outweigh everything. The dominance is going to trump over the normal red-green color blindness. I'm actually going to, it's all in the slides, but I prefer to draw it out. So I'm going to draw it out again. So a female has 2 X chromosomes, correct? On these X chromosomes, she has the ability to see, I'm just going to say color. Okay, so we have big C, okay? If there's 2 dominant colors, she can see color. If she has 1 big C and 1 little c, the dominant is going to override and trump the recessive, so she can still see color. Okay, she can still see color, red-green color. If the female has little c and another little c, she will not be able to see color. So hopefully this makes sense to you guys, right? Now, when we look at a male, males do not have 2 X chromosomes. They have 1 X chromosome and 1 Y chromosome, okay? So this X chromosome holds the gene to be able to see the red-green color blindness. This Y chromosome doesn't. It's just basically empty space. It's in this particular case, it's kind of useless. So if the male has 1 big C, they can see the color. And if the male has 1 little c, they're pretty much screwed and they cannot see color because the Y does not hold a big C because the Y chromosome is a completely different chromosome. And so they don't have another slot. See, if we have 1 little, like by we, I mean female, sorry. If females have 1 little c, we can always fall back on our other X chromosome to see if there's a big C there, and therefore if there is, we can see color. If there isn't, then we don't see color, right? But we at least have something to kind of fall back on. Males don't have anything to fall back on because their Y chromosome is completely different from the X chromosome. It does not have the same genes. These 2 chromosomes are not homologous to each other. So when the male has little c, they have nothing to fall back on, and so because they only have a Y chromosome. And so whatever the male gets, whatever X chromosome they get, whatever gene they get on the X chromosome, even if it's just 1 allele, that's what they will be. That's what they will have. And usually, sometimes it's not a terrible thing, like being red-green colorblind. It's not really a terrible thing. Yes, you can see the colors red and green, but it's not exactly terrible. However, there can be other really bad things like hemophilia. Hemophilia is a sex-linked recessive trait. So again, you need to have 2 recessive traits, 2 recessive genes, 2 recessive alleles to have hemophilia, or basically this ability not to clot your wounds, and so basically bleed to death. Not good, right? This particular gene is found on the X chromosome. So for females, in order for them to be hemophilic, they need to have 2 recessive alleles. For males to be hemophilic, they need to have 1 recessive allele. So it's really, really easy, and much easier and much more common for males to have these specific X-linked disorders, right? So if we go back to the example of red-green color blindness, or even if we want to do the hemophilia, say we have a female, that's big C, little c. Okay, I'm going to draw a Punnett square that comes together with a male, X and Y. And this male, this is a Y, sorry, X and Y. This male can also see color. It's a big C. Okay, so their possible offspring, they can, this female, she gets one X from the dad and one X from the mom. The one X from the dad has a cap big C, and this one X from the mom has a big C. She can see color. She's all good. This female right here, she gets one X from the dad with the big C, one X from the mom with the little c. But even though we have a little c, we still have this big C is going to dominate over it, so she can still see color. However, in this case right here, she is a carrier, right? How about here with the males? With the males, the possible male offspring, you get one X from the mom, in this case it's a big C, and then the Y from the dad. So this is going to be a male, and he can see color, okay? But in this case right here, he gets the X from the mom with the little c, and the Y from the dad. And so because of that, this male is going to be red-green colorblind. So you can see that the females, there's 100% of them, both of them, all females that come about from this particular mating are going to be able to see the color, no problem. But all males that come about, all male offspring that come about from this particular mating, 50% of them will not be able to see the red-green color, 50% of them will. And it's the same thing with hemophilia. Again, this just shows and can expound the fact that males are more prevalent, are more capable of basically taking up these genes just because they don't have another slot to basically kind of fall back on, right? And so sometimes it's not as bad because it's red-green color blindness is not really that bad of a thing. My dad has it. My uncle has like total color blindness. I think my grandma actually has it as well, right? So it's not really that bad of a thing. It may be a, somewhat of a disadvantage, but it's not an actual terrible disorder. While there are other things like Duchenne muscular dystrophy or hemophilia, these are all genes that are found that are sex-linked, that are found on your X chromosome, and males get the short end of the stick essentially, and they have a higher rate of getting these particular disorders because once they have one recessive allele, they can't fall back on anything else. They only have a Y chromosome. They don't have another X chromosome to fall back on to basically counteract that recessive allele. So that is it for chapter 9, and I shall see you guys in the next video. Bye, guys.